Chapter 2 of the podcast series, Biological Classification. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific, but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, a two-kingdom system of classification with Plantae and Animalia kingdom was developed that included all plants and animals respectively. This system did not distinguish between the eukaryotes and the prokaryotes, unicellular and multicellular organisms, and photosynthetic green algae and non-photosynthetic fungi organisms. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two-kingdom system of classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships, etc. Classification systems for the living organisms have hence undergone several changes over the time. Though plant and animal kingdom have been a constant under all different systems, the understanding of what groups organisms will included under this kingdom have been changing. The number and nature of the other kingdoms have also been understood differently by different scientists over the time. I am going to read the chart which is given in the table 2.0 and the characteristics of the five kingdoms. First, I am going to read about the Monera, whose cell type is prokaryotes, and Monera cell wall is non-cellulosic polysaccharide plus amino acid, and nuclear membrane is absent. Nuclear membrane is absent and body organization is cellular. Mode of nutrition is autotrophic, chemosynthetic and photosynthetic, and heterotrophic, saprophytic and parasitic. Second, we are going to read about Protista. Cell type is eukaryotic and cell wall is present in some and nuclear membrane is present. Cellular is the body organization and mode of nutrition is autotrophic, that is photosynthetic and heterotrophic. Next, fungi. Cell type is eukaryotic, cell wall is present with chitin, nuclear membrane is present, body organization is multicellular with loose tissue, and mode of nutrition is heterotrophic, saprophytic and parasitic. Next is Plantae, whose cell type is eukaryotic, cell wall is present with cellulose, and nuclear membrane is present, body organization is tissue organ, and mode of nutrition is autotrophic, photosynthetic. And Animalia, cell type is eukaryotic, cell wall is absent in the kingdom Animalia, and nuclear membrane is present, and body organization is tissue, organ and organ system, and mode of nutrition is heterotrophic, that is holozoic and saprophytic. This is the table I have read. Okay. Next, we are proceeding to the paragraph. R.H. Whittaker in 1969 proposed a five-kingdom classification. The kingdom defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationship. Table 2.1 gives a comparative account of different characteristics of the five kingdoms. The three-domain system has also been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryotic kingdoms in the three-domain and thereby a six-kingdom classification. You will learn about this system in details in the higher class. Let us look at this five-kingdom classification to understand the tissue and consideration that influenced the classification system. Earlier classification system included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms and the angiosperms under plants. The character that unified this whole kingdom was that all the organisms included had a cell wall in their cells. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae cyanobacteria with other groups which were eukaryotic. It also grouped together the unicellular organisms and the multicellular ones, say for example Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group fungi and the autotrophic green plants, though they also showed a characteristic difference in their walls composition. The fungi had chitin in their walls, while the green plants had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in separate kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in the kingdom Protista. Kingdom Protista has brought together Chlamydomonas, Chlorella, earlier placed in the algae with plants and both having cell walls, with Paramoecium and Amoeba which were placed in the kingdom Animal which lacks cell wall. It has put together organisms in earlier classification were placed in different kingdoms. This happened because the criteria for classification has changed. This kind of changes will take place in future too, depending on the improvement in our understanding of characteristics and evolutionary relationships. Over the period of time, an attempt has been made to evolve a classification system which reflects not only the morphological, physiological and reproductive similarities, but is also phylogenetic, that is based on evolutionary relationship. In this chapter, we will study characteristics of kingdom Monera, Protista and fungi of the Whittaker system of classification. The kingdom Plantae and Animalia, commonly referred to as plant and animal kingdom respectively, will be dealt separately in chapter 3rd and 4th. Kingdom Monera. Bacteria are the sole members of the kingdom Monera. They are the most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot springs, deserts, snow and deep oceans, where very few other life forms can survive. Many of them live in or in other organisms as parasites. Bacteria are grouped under 4 categories based on their shape: the spherical coccus, that is plural cocci, the rod-shaped bacillus, plural bacilli, the comma-shaped vibrium, plural vibrio, and the spiral spirillum, plural spirilla. Though the bacterial structure is very simple, they are very complex in behavior. Simple in structure and complex in behavior. Understood? Compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrates. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs, that is, they depend on other organisms or on a dead organic matter for food. Archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extreme salty areas halophiles, hot spring thermoacidophiles, and marshy areas methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminants, animals such as cows and buffaloes, and they are responsible for the production of methane gas, biogas from the dung of these animals. We are going to read the eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of a rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to as blue-green algae, have a chlorophyll A similar to green plants and are photosynthetic autotrophs. The cyanobacteria are unicellular, colonial or filamentous, freshwater marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. They often form bloom in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts. Example, nostoc and anabina. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, nitrites, and ammonia and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume roots, etc. Some are pathogens causing damage to human beings, crops, farm animals, and pets. Cholera, typhoid, tetanus, citrus canker are well-known diseases caused by bacteria. It's an important line. Cholera, typhoid, tetanus, citrus canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission. Sometimes, under unfavorable conditions, they produce spores. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to the other. The mycoplasma are the organisms that completely lacks a cell wall. They are the smallest living cells known and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. Kingdom Protista we are going to read about. All single-celled, again I am repeating it, all single-celled eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protista to a one biologist may be a plant to another. In this book, we include chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans under Protista. Members of Protista are primarily aquatic. This kingdom forms a link with the other dealing with plants, animals, and fungi. Being eukaryotes, the Protistan cell bodies contain a well-defined nucleus and other membrane-bound organelles. Being eukaryotes, the Protistan cell body contains a well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. Protists reproduce asexually and sexually by a process involving cell fusion and zygote formation. Reproduce asexually and sexually by a process involving cell fusion and zygote fusion. Cell fusion and zygote formation. means by a special process. Self Chrysophytes. These groups include diatoms and golden algae. That's they are found in fresh water as well as in marine environments. They are microscopic and float passively in water currents plankton. Most of them are photosynthetic. In diatoms, the cell wall forms two thin overlapping shells which fit together as in soap box. The walls are embedded with silica and thus the walls are indestructible. Thus, diatoms have left behind large amount of cell walls deposits in their habitat. This accumulation over billions of years is referred to as diatomaceous earth. Being gritty, this soil is used in polishing, filtration of oils and syrups. Diatoms are the chief producer in the ocean. Next, we are going to read about the dinoflagellates. सुन रहे हो? These organisms are mostly marine and photosynthetic. Marine and photosynthetic. They appear yellow, green, brown, blue or red depending on the main pigments present in their cells. Their cell walls has stiff cellulose plates on the outer surface. Most of them have two flagella, one lies longitudinally and the other transversely in a furrow between the wall plates. Very often red dinoflagellates, example Gonia lux, undergo such rapid multiplication that they make the sea appear red, that is red tides. Toxins released by such large numbers may even kill other marine animals such as fishes. Next, we are going to read about euglenoids. Majority of them are freshwater organisms found in stagnant water. Instead of cell wall, they have a protein-rich layer called a pellicle, which makes their body flexible. They have two flagella, a short and a long one. Though they are photosynthetic in the presence of sunlight, when they deprived of sunlight, they behave like heterotroph by predating on other smaller organisms. Interestingly, the pigment of euglenoid are identical to those that present in the higher plants. Example, euglena. Next, we are going to read about the slime molds. Slime molds are saprophytic protists. The body moves along decaying twigs and leaves engulfing organic material. Under suitable conditions, they form an aggregation called plasmodium, which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiates and forms fruiting body. Unfavorable condition में fruiting body बनाता है, bearing spores at their tips. The spores possesses true walls. They are extremely resistant and survive for many years, even under adverse conditions. The spores are dispersed by air currents. Next, we are going to read about protozoans. All protozoans are heterotrophs. And live as predators or parasites. They are believed to be primitive relatives of animals. There are four major groups of protozoans. First is amoebic protozoans. These organisms live in fresh water, seawater or moist soil. They move and capture their prey by putting out pseudopodia, false feet as in amoeba. Marine forms have silica shells on their surface. Some of them, such as entamoeba, are parasites. Next, flagellated protozoans. The members of this group are either free-living or parasitic. They have flagella. The parasitic forms causes disease such as sleeping sickness. Example, trypanosoma. Ciliated protozoans. These are aquatic, actively moving organisms because of the presence of thousands of cilia. They have a cavity gullet that opens to the outside of the cell surface. The coordinated movement of rows of cilia causes the water laden with food to be stirred into the gullet. Example, paramoecium. Next, sporozoans. This includes diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is Plasmodium malarial parasite, which causes malaria, a disease which has a staggering effect on human population. Kingdom fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. You must have been seeing fungi on a moist bread and rotten fruits. The common mushroom you eat and toadstools are also fungi. White spots seen on mustard leaves are due to the parasitic fungus. Some unicellular fungi, example yeast, are used to make bread and beer. Other fungi causes diseases in plants and animals. Wheat rust causing Puccinia is an important example. Some are the source of antibiotic, example penicillium. Fungi are cosmopolitan and occur in air, water, soil and on animals and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in the refrigerator? Yes, it is to prevent food from going bad due to bacterial or fungal infection. With the exception of yeast, which are unicellular, fungi are filamentous. Their body consists of long, slender, thread-like structure called hypha. The network of hypha is known as mycelium. Some hypha are continuous tube filled with multinucleated cytoplasm. These are called zoonotic hypha. Others have septa or cross walls in their hypha. The cell walls of fungi are composed of chitin and polysaccharides. It's an important line. Most fungi are heterotrophic and absorb soluble organic matter from dead substrates and hence are called saprophytes. Those that depend on living plant and animals are called parasites. They can also live as symbiont in association with algae lichens and with roots of higher plant as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission and budding. Asexual reproduction is by spores called conidia or sporangiospores or zoospores. And sexual reproduction is by oospores, ascospores and basidiospores. The various spores are produced in distinct structure called fruiting body. The sexual cycle involves the following three steps. Fusion of protoplasm between two motile or non-motile gamete is called plasmogamy. Fusion of two nuclei is called karyogamy. Third, meiosis in zygote resulting in haploid spores. When a fungus reproduces sexually, two haploid hypha of compatible mating types come together and fuse. In some fungi, the fusion of two haploid cells immediately results in diploid cells 2N. However, in other fungi, Ascomycetes and Basidiomycetes, an intervening dikaryotic stage N plus N, that is 2 nuclei per cell, occurs. Such a condition is called a dikaryon and the phase is called dikaryophase of fungus. Later the parental nuclei fuse and the cells become diploid. The fungi form fruiting bodies in which reduction division occurs, leading to formation of haploid spores. The morphology of the mycelium, mode of spore formation and fruiting body form the basis for the division of the kingdoms into various classes. Understood? First, Phycomycetes. Members of Phycomycetes are found in aquatic habitats and on decaying wood in moist and damp places or as obligate parasites on plants. The mycelium is aseptate and zoonocytic. Asexual reproduction takes place by zoospores, that is motile, or by apollinospores, that is non-motile. Zoospore is motile and apollinospore is non-motile. These spores are endogenously produced in sporangium. A zygospore is formed by fusion of two gametes. These gametes are similar in morphologies, isogamous, or dissimilar, that is anisogamous or oogamous. Some common examples of Phycomycetes are mucor, rhizopus, the braid mold mentioned earlier, and the albugo, the parasitic fungi on mustard. Second, Ascomycetes, commonly known as sac fungi. The Ascomycetes are mostly multicellular, example penicillium, or rarely unicellular, example yeast, that is Saccharomyces. They are saprophytic, decomposer, parasitic, or coprophilous, growing on dung, means gobar. Mycelium is branched and septate. The asexual spores are conidia produced exogenously on the spatial mycelium called conidiophores. Conidia on germination produce mycelium. Sexual spores are called ascospores, which are produced endogenously in a sac-like asci, singular ascus. These asci are arranged in different types of fruiting bodies called ascocarps. Some examples of Ascomycetes are Aspergillus, Claviceps, and Neurospora. Neurospora is used extensively in the biochemical and genetic work. Many members like morels and truffles are edible and are considered as delicious. Swadist. Next is Basidiomycetes. Commonly known form of Basidiomycetes are mushroom, bracate fungi, or puffballs. They grow in soil, on logs and tree stumps, and in living plant bodies as parasites, example rust and smuts. The mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains or genotypes. The resultant structure is dikaryotic, which ultimately give rise to basidium. Karyogamy and meiosis take place in the basidium, producing 4 basidiospores. The basidiospores are exogenously produced on the basidium, plural basidia. The basidia are arranged in fruiting bodies called basidiocarps. Some common members are agaricus, that is mushroom, eustilago, smut, and Puccinia is the rust fungus. Next is Deuteromycetes, commonly known as imperfect fungi because only the asexual or vegetative phases of these fungi are known. When the sexual forms of these fungi were discovered, they were moved into classes they rightly belong to. It is also possible that the asexual and vegetative stages have been given one name and placed under Deuteromycetes, and the sexual stage another and placed under another class. Later, when the linkages were established, the fungi were correctly identified and moved out of the Deuteromycetes. Once perfect sexual stage of members of Deuteromycetes were discovered, they were often moved to Ascomycetes and Basidiomycetes. The Deuteromycetes reproduce only by asexual spores known as conidia. The Deuteromycetes reproduce only by asexual spores known as conidia. The mycelium is septate and branched. Some members are saprophytes or parasites, while a large number of them are decomposer of litter and help in mineral cycling. Some examples are Alternaria, Coletrichum and Trichoderma. Alternaria, Coletrichum and Trichoderma. Kingdom Plantae. Kingdom Plantae includes all eukaryotic chlorophyll containing organisms commonly called plants. A few members are partially heterotropic such as the insectivorous plants or parasites. Bladderwort and Venus flytrap are examples of insectivorous plant, and Cuscuta is a parasite. The plant cells have an eukaryotic structure with prominent chloroplast and cell wall mainly made of cellulose. We will study the eukaryotic cell structure in detail in chapter 8. Plantae include algae, bryophytes, pteridophytes, gymnosperm and angiosperm. The life cycle of plant has two different distinct phases, the diploid sporophytic and the haploid gametophytic, that alternate with each other. The length of the haploid and diploid phases and whether these phases are free living or dependent on other vary among different groups in plants. This phenomena is called alternation of generation. We will study further details in the kingdom in the chapter 3rd. Kingdom Animalia. This kingdom is characterized by heterotropic eukaryotic organisms that are multicellular and their cells lack cell walls that directly or indirectly depend on plant for food. They digest their food in an internal cavity and store food reserve as glycogen or fat. Their mode of nutrition is holozoic by ingestion of food. They follow a definite growth pattern and grow into adults that have a definite shape and size. Higher forms show elaborate sensory and neuromotor mechanisms. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female followed by embryological development. Salient features of various phyla are described in chapter 4. Now we are going to read about viruses, viroids, prions and lichens. There are many chances of coming questions from here. In the 5 kingdom classification of Whittaker, there is no mention of lichens and some acellular organisms like viruses, viroids and prions. These are briefly introduced here. All of us who have suffered the ill effects of common cold or flu know what effects viruses can have on us. Even if we do not associate it with our condition, viruses did not find a place in classification since they are not considered truly living. If we understand living as those organisms that have a cell structure, the viruses are non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. होस्ट की वाट लगा दो. Would you call viruses living or non-living? Virus means venom or poisonous fluid. Dmitry Ivanovsky in 1892 recognized certain microbes as causal organisms of the mosaic disease of tobacco. These were found to be smaller than bacteria because they passed through bacteria-proof filters. M.W. Bazarnik in 1898 demonstrated that the extract of the infected plants of tobacco could cause infection in healthy plants and named the new pathogen virus and called the fluid as Contagium vivum fluidum, infectious living fluid. W.M. Stanley in 1935 showed that viruses could be crystallized and crystals consist largely of proteins. They are inert outside their specific host cells. Viruses are obligate parasites. In addition to proteins, viruses also contain genetic material that could be either RNA or DNA. No viruses contain both RNA or DNA. A virus is nucleoprotein and the genetic material is infectious. In general, viruses that infect plant have a single-stranded RNA, and viruses that infect animals have either single or double-stranded RNA or double-stranded DNA. Bacterial viruses or the bacteriophage, that is, virus that infect the bacteria, are usually double-stranded viruses. The protein coat called capsid, made of a small subunit called capsomers, protect the nucleic acid. These capsomers are arranged in helical or polyhedral geometric forms. Viruses cause diseases like mumps, smallpox, herpes, and influenza. Diseases like mumps, smallpox, herpes, and influenza are caused by viruses. AIDS in human is also caused by a virus. In plants, the symptom can be mosaic formation of leaf rolling and curling, yellowing and vein clearing, dwarfing and stunted growth. Symptoms can be mosaic formation, leaf rolling and curling, yellowing and vein clearing, dwarfing and stunted growth. Viroids. In 1971, Theo Dinner discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber diseases. It was found to be a free RNA. It lacked the protein code that is found in viruses. Viroid are without protein code and hence the name viroid. The RNA of the viroid was of low molecular weight. Next is prions. In modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded protein. The agent was similar in size to viruses. These agents were called prions. The most notable diseases caused by prions are bovine spongiform encephalopathy, that is BSE, commonly called mad cow disease in cattle, and its analogous variant Chiari-Jacob disease, that is CGD, in humans. Lichens. Lichens are symbiotic association, that is mutually and useful association between algae and fungi. The algal component is known as phycobiont and the fungal component is known as mycobiont, that you can remember by fungal is mycobiont, you can remember it by FM, which are autotrophic and heterotrophic respectively. That is, algal component is phycobiont, which is autotrophic, and fungal component mycobiont is heterotrophic. Algae prepare food for fungi and fungi provide shelter and absorb mineral nutrients and water for its partner. So close is their association that if one saw a lichen in nature, one would never imagine that they had two different organisms within them. Lichens are very good pollution indicators. They do not grow in a polluted area. Understood? I am going to read the summary in a speedy and a quick way. Okay? Biological classification of plant and animal were first proposed by Aristotle on the basis of simple morphological characters. Linnaeus later classified all living organisms into two kingdoms, Plantae and Animalia. Whittaker proposed an elaborate five-kingdom classification, Monera, Protista, Fungi, Plantae and Animalia. The main criteria of the five-kingdom classification were cell structure, body organization, mode of nutrition and reproduction, and phylogenetic relationship. This is important, right? The main criteria for five-kingdom classification is cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationships. In the five-kingdom classification, bacteria are included in kingdom Monera. Bacteria are cosmopolitan in distribution. This organism show the most extensive metabolic diversity. Bacteria show most extensive metabolic diversity. Bacteria may be autotrophic or heterotrophic in their mode of nutrition. Kingdom Protista include all single-celled eukaryotes such as cryophyte, dinoflagellate, euglenoid, slime molds, and protozoans. Protista have defined nucleus and other membrane-bound organelles. They reproduce both asexually and sexually. Members of kingdom fungi show a great diversity in structure and habitat. Most fungi are saprophytic in their mode of nutrition. They show sexual and asexual reproduction. Phycomites, ascomites, basidiomytes, and deuteromycetes are the 4 classes under this kingdom. The plant include all eukaryotic chlorophyll-containing organisms, that is, algae, bryophytes, pteridophytes, gymnosperm, and angiosperm are included in this group. The life cycle of plant exhibit alternation of generation, gametophytic and sporophytic, The heterotrophic eukaryotic multicellular organisms lacking a cell wall are included in the kingdom Animalia. The mode of nutrition of this organism is holozoic of the kingdom Animalia. They reproduce mostly by the sexual mode. Some acellular organisms like viruses and viroid as well as the lichens are not included in the 5 kingdom system of classification. With this, our chapter is completed. Thank you for hearing the podcast. Have a wonderful